Hello students, myself Aparna, Botany faculty from MS Medipatnam IOE. Today I am going to describe about the easy point of learning for the TS Board of Intermediate Botany Practical Examinations. Very easily make yourself comfortable and confident before going to the examinations by observing each and every slide of this whatever the video we are preparing. It helps you a lot. Let us start our session with what kind of questions, how many questions they are going to give, and how many marks they are going to be allotted, and what you need to write in each and every session. Everything we will be discussing very detailed manner. Let's start our video. First, we have totally 3 main sessions. First one is, first question is about describing the given family or twig. They will be giving certain twig to you. You need to find it out, what kind of vegetative characters and floral characters, and after seeing those we need to find out the family, and after that you need to write the description about floral formula, floral diagram, and the label diagram of twig with inflorescence as well as LS of flower. So, here they have mentioned each and every point how many marks are going to be allotted. And for example, totally we have 3 families in our syllabus, that is Fabaceae, Solanaceae and Liliaceae. So, let us concentrate on Fabaceae, how the twig will be, and here Tephrosia, this by observing this twig, and this is pinnate compound, and here you can see that papillonaceous corolla. By observing this twig you can find that very easily this is belongs to Fabaceae. And let us see other twig. Suppose if you are getting this kind of twig by observing flower as well as the leaf with reticulate venation, you can find this inflorescence is solitary cymose. So, by observing this we can find this twig belongs to Solanaceae member. And we have another things, the last family of monocot. By observing this, these are spring onions, they will be giving this kind of twigs to you. So, here fibrous root system is there and storage leaves are there, and the leaves are with parallel venation. So, by finding this you can find very easily, by observing this you can say this is belongs to Liliaceae family. So, after this you need to immediately draw the twig diagram with inflorescence for that half mark, and after that by based on what vegetative characters you are saying are identifying this particular twig so and so family, that is based on vegetative characters you have to. Then we need to find the floral characters also. So, that means calyx, corolla, androecium, gynoecium. By everything we need to describe that in your examination paper. For that we have 2 marks. So, let us see, these are all together for make you very comfortable with regarding the practical examination point of view. We have taken most important points. If you write this, this is more than enough to get good marks here. So, Fabaceae and Solanaceae, Liliaceae all together in a comparative degree, if you learn the things, it is very easily you can remember within short span of time. So, check it out. The first one, vegetative characters, you need to write about habitat. Fabaceae is mesophyte, Solanaceae also mesophyte, and Liliaceae also mesophyte. Habit is annual herb. It might be Solanaceae is annual herb or shrub, if it is Liliaceae perennial herb. If you are observing the root system of Fabaceae and Solanaceae, both are taproot system because they belongs to dicot. And if you are observing Liliaceae, that root system is fibrous root system, and if you are observing the stem of this aerial, erect, cylindrical, herbaceous, and if you are observing the same characters in Solanaceae, but it is hairy. In Liliaceae, underground stem modification you can see, that is bulb. And if you are observing leaf, it is in Tephrosia, pinnately compound leaf, petule is present, alternate phyllotaxy you can see, and very importantly these are stipulate, other two families are exstipulate, and the venation being a dicot reticulate venation. And if it is Solanaceae, simple leaf, petulate, alternate phyllotaxy, exstipulate with reticulate venation. If you are observing the same thing about the leaf, in the case of Liliaceae, simple leaf, linear, sessile, radical leaf, exstipulate with alternate phyllotaxy and parallel venation. This is these are about the vegetative characters. So while any one of the family will be given to you, so you have to consider any one of the vertical column while writing the examination point of view. You need to take either Fabaceae, Solanaceae or Liliaceae based on what you got a kind of twig in examination hall. Okay. So next one is floral characters. For these you are going to get 2 marks. First one about the general description of the flower. If you are observing Fabaceae members, they are bracteate, bracteolate, complete bisexual, zygomorphic, pedicellate, perigynous, and flower is pentamerous. And if you are observing in Solanaceae, ebracteate, complete flower, bisexual, here it is actinomorphic flower, and pedicellate, hypogynous, it is also pentamerous flower. If it is Liliaceae member, bracteate, complete, bisexual, actinomorphic, and pedicellate, hypogynous, but this is trimerous flower. First 2 families are pentamerous, but Liliaceae member, Allium cepa is trimerous. Calyx is 5 sepals which are fused velvet aestivation, gamosepalous condition in Fabaceae. If it is Solanaceae, sepals are 5, those are also fused gamosepalous, velvet aestivation, the calyx is persistent calyx. If you are observing corolla in these 2 members, first in Fabaceae, 5 petals are there, those are not fused, and they are in papillonaceous corolla, 1 standard petal toward posterior, 2 lateral wing petals in turn fused with 2 keel petals. This kind of aestivation is called vexillary aestivation observed in Fabaceae. If it is in Solanaceae, petals are fused, gamopetalous, funnel shaped, twisted aestivation is present. And in the case of Liliaceae, there is no separation of calyx and corolla. We can find tepals as a perianth members. Here, totally 6 tepals are arranged in 2 holes, outer hole or tepal anterior and inner hole or tepal posterior. The kind of aestivation in Liliaceae is velvet aestivation. Now, let us concentrate on andrecium. 10 stamens are there, 9 are fused and 1 is free. That condition is called diadelphous condition, and this or anthers are dithecous, basifixed, introsed and longitudinal dehiscence. If you are observing in Solanaceae members, stamens are 5 which are fused with petals. That condition is called epipetalous condition, and anther is dithecous, basifixed and longitudinal dehiscence. This point is common to all the 3 formulas. And now Liliaceae, 6 stamens are there, 3 are in one hole and other 3 are in other hole, and these are stamens are attached to the tepals. This condition is called epiphyllous condition. Here also anther dithecous, intros, basifixed and longitudinal dehiscence. Now let us concentrate on gynoecium. And gynoecium in Fabaceae, only single corpel is present. So monocorpulary, unilocular with a marginal placentation, ovary is half superior and style terminal, stigma capiteate. And if you are observing in Solanaceae, 2 carpels are there, so it is bicarpellary tetralocular superior ovary, solen axil placentation, and ovules are arranged with an angle of 45 degrees, style terminal, stigma capitate. This is the common point between all the 3 families. Now Liliaceae, it is tricarpellary trilocular syncarpal superior ovary with axile placentation. You can check it out. And fruit, in the case of Fabaceae, it is legume. In the case of Solanaceae, it is capsule, and in the case of Liliaceae also it is capsule. These all information regarding the twig what they have provided. First, Fabaceae member, we are talking about all this description about Tephrosia. For Solanaceae member, we are talking all this description about Datura. For Liliaceae, we are taking Allium cepa. We are observing all these characters in these twigs given to you. And these are the above provided information is giving 3 marks to us, vegetative characters 1 mark and floral characters 2 marks. Out of 6 marks, you are getting 3 marks. Right now, you need to focus on this. Very particularly, they are giving marks, allotment of marks is very particular. For twig with inflorescence, we are getting half mark, and for Elisa flower, we are getting half mark. Floral diagram, we are getting half mark. For identification of family, we are getting 1 mark, and for floral formula, we are getting half mark. Overall, for this particular representation, you are getting 3 marks, and above description 3 marks. Totally, we are getting 6 marks for this session 1. So, these are the diagrams. Mandatorily, everybody should learn how to draw all the 3 diagrams what they have provided here. So, for this Solanaceae, that is for Liliaceae. This is about the families. Very keenly you need to learn the things. Very limited information we have provided. So, this is very easy to learn also. Now, let us concentrate on TS of given material. Transverse section you need to cut, and here we have 4 different kinds of section cuttings. Those are monocot stem, dicot stem, monocot root and dicot root. And here very importantly, how you are cutting the section, that for that very particularly preparation of slide they are giving 3 marks. And for identification after preparation and after placing that slide under microscope, what you have identified, for that they are giving 1 mark. Then after seeing that slide under microscope, you get an imaginary diagram of that sector only you need to draw. For that drawing of diagram of section cutting, of transverse section cutting, we are getting 2 marks. Overall for this second question we are getting 6 marks here. Now here also for identification characters we made very easy to the students to learn in a comparative orientation. And here first one is dicot root identification character. We need to compare both the roots and we need to compare both the stems together, then we can learn it very easily. Suppose first 2 columns about the root, let us concentrate on this. Both are containing root hairs. Monocot root is also containing root hairs and dicot root also containing root hairs. Now epidermis being a root, it is not containing any cuticle, that is why it is uncutinized. And here if you are observing the diagram, cortex is, this is the diagram of this is cortex and this is stele. Cortex is very larger when compared to the stele. This is the common point in both monocot and dicot root. And here if you are observing that the kind of vascular bundles in both monocot root and dicot root as an identification characters, both are radial vesicular bundles they have, and xylem is exarch. You are very much aware of that. If protoxylem faces towards, see the diagram very carefully, the smaller xylems are called protoxylems. If this protoxylem faces towards the pericycle, then that is called exarch xylem. You need to draw the diagram very clearly here. See this, I am enhancing the size of the diagram. So, here big size xylem should be inside and small size xylem should be towards the pericycle. Here 4 xylems you can see in dicot root, that is why it is tetrarch. If you are observing this monocot root, here I am counting it 1, 2, 3, 4, 5, 6, 7. Here totally you can find 8 xylems. This kind of things is called polyarch. We are calling this as polyarch xylem. This is the major difference between dicot root and monocot root. Here very minute pith or the center medulla is observed, but here well developed pith or medulla is seen in the case of monocot root. This is the great difference between the monocot root and dicot root. Here tetrarch xylem in dicot, there polyarch xylem in dicot. Here pith is minutely developed, here pith is well developed. Now let us see the other characteristic properties here. Now just now we have discussed about medulla, poorly developed and well developed, and by observing all these following characters we can differentiate monocot and dicot root, as well as if the root is there we can find root hairs. Instead of stem we can find this with very particularly exarch xylem make you to differentiate from root and stem. Now let us concentrate on other two, dicot stem and monocot stem. These are identification characters. If it is in the case of dicot stem, trichomes are present. If it is monocot stem, trichomes are absent. And in both the cases of stem, epidermis contain cutin, and whether it is monocot stem or dicot stem. Hypodermis is collenchymatous in the case of dicot stem, and hypodermis is sclerenchymatous in the case of monocot stem. And if you are observing the vesicular bundles, very clearly you can differentiate dicot stem and monocot stem. Dicot stem have ring shaped vesicular bundles and monocots have scattered vesicular bundles. Let us see the diagram of that. So, these are ring like structure it is forming in the case of dicot stem. The vesicular bundles are scattered or diffused throughout the ground tissue. This is the great difference between dicot stem and monocot stem. And if you are observing, in both the cases xylem is endark, and xylem in the case of monocot stem, xylem vessels are Y-shaped. And endodermis have starch sheath in dicot stem, pericycle have semi-lunar patches of sclerenchyma and collenchyma. Endodermis and pericycle are completely absent in the case of monocot stem. And if you are observing the vesicular bundles of monocot stem and dicot stem, let us see. Dicot stem, the vesicular bundles are conjoined collateral open type because they have cambium. And if it is in the case of monocot stem, vesicular bundles are conjoined collateral closed. And if you are observing secondary growth, that will be observed in dicot stem because of presence of cambium. And if you are observing monocot stem, vesicular bundles will not have the cambium, that is why there is no secondary growth. By observing above characters, we can happily say that the given session cutting is whether dicot stem or monocot stem. Very importantly, here well developed medulla is present, there no medulla is observed. Here pericycle ring shaped vesicular bundles, there scattered vesicular bundles can be observed. So, these are the diagrams. Mandatorily you need to draw this. For this drawing you are going to get 2 marks, and the overall description of session second question, totally 6 marks this question contains, and 3 marks for session cutting and 3 marks, 1 mark is for identification and 2 marks for drawing these diagrams. So, based on what you get, that kind of diagram you are going to draw in your examination. Okay. So, how to do the session cuttings? Very keenly we need to check that how thin it should be. Let us see that session cutting.
Ready.
Slide. Bring the microscope on. Cover slip is not there. You just put another slide. After session cutting, we are going to concentrate on next last one. It's live experiments here. So, here we have totally 4 live experiments are there. First one is study of osmosis by potato osmoscope. Dear students, you please remember, you need not to worry about that. You need to understand the live experiment very clearly. Here you need to write only aim, principle, observation and inference or result. Okay. So, you need not to write any requirements or procedures, something like only aim, principle, observation and result, that only you need to mention. So, let us concentrate on our first experiment, study of osmosis by using potato osmoscope, and you are all aware of potato here. So, I have taken normal potato. After that I peel this potato with a peeler, then I made it into half. After that I placed a small well inside this potato. So, now in this deep well I am going to place sugar solution. This is a sugar solution, and I am going to place till to the half of the level of the sugar solution in this well, and I placed small pin till to that half level of the sugar solution. So, now I placed this in a petri dish containing dilute water. I am pouring normal water in this petri dish. So now you can see only half of this well I filled with a concentrated solution, that is, when compared to this is concentrated sugar solution I have taken. Now I am placing that well of potato which has half filled with sugar solution. For that half filling I placed one pin. Now I am going to keep this for half an hour. Now from this, what you are going to observe after half an hour? So once again I repeat this experiment. First I have taken a potato and I peel that. After peeling I make the potato into half, and then I made a well. In that well, half of the well is filled with sugar solution. After filling with sugar solution, I put a mark by placing pin. This is the pin for half of the sugar solution. That means leftover half is empty right now. Now I placed this in a petri dish containing dilute solution or water. After half an hour, you are going to observe, or as an inference, the water level from the dip is moving about to the pin, or sometimes if you are leaving for certain duration of time, it will be overfloating also. What absolutely happening is movement of water or solution, movement of water or solvent from the region of low concentrated solution, that means petri dish, to the high concentrated solution, that is inside the well sugar solution, and through a selectively permeable membrane. Here this potato is acting as a selectively permeable membrane. Once again, what you are going to observe after half an hour, the water is moving from the low concentrated outside solution to the high concentrated solution inside the depth. Now itself I am going to see there is a rise in sugar solution, it is coming about to the pin. So you can also very easily identify that. This is about potato osmoscope by osmosis by potato osmoscope. Okay. Now let us concentrate on our second lively experiment or physiological experiment. Let us see. Plasmolysis. Second experiment, here also I am mentioning only aim, and we are taking here, this experiment is about the plasmolysis in epidermal peel of rhodiscolor leaves. This is the rhodiscolor leaf, and you can see very easily backside a little bit purple color is there and here green color is there. So now we are going to remove this epidermal peel. How to remove that epidermal peel? Very easily, if you bend this and if you are tearing this leaf like this, very easily it will come. A little minute peel is coming here. You just cut it and make sure it off. Green color should not be there at the epidermal peel, only purple color should be there. Now I am placing this in a petri dish containing hypertonic solution. You just check it. Earlier itself I peeled this rhodiscolor leaves and you can check it out. For a while I have placed this epidermal peel of this leaf in a solution containing with high concentration, that is called hypertonic solution. Now if you are taking this leaf and placing that on slide and observing under microscope, there is a shrinkage in the cells of leaf because of hypertonic solution in this. We are placing the epidermal peel in hypertonic solution. When compared to the cytoplasm of the cell, the concentration of the solution is outside more when compared to inside. So, here water is coming from inside the cell to the outside the cell, that is called exoosmosis. This leads to the shrinkage of the cell, that is called plasmolysis. Once again I am repeating that. First we need to take this leaf and we need to peel it off this only purple color epidermal peel, then we need to place that in hypertonic solution. After half an hour, if you observe under microscope by placing this epidermal peel on slide, there will be a shrinkage in cells because of exoosmosis. So, here the water loses out from the cell to the external medium because external medium is hypertonic. So, this is about the second experiment of plasmolysis of epidermal peel of leaf. One second. This must be. Third experiment, cobalt chloride method in order to find out the transpiration by leaf stomata. So, here this is very easily you can as of now whatever the thing I have I am going to explain to you. So, before they start their experiment, if you are getting live experiment of transpiration, finding out transpiration by stomata, they will be giving one plant to you. To that plant, you have to put by using slide and cover slip, at least cover slip, you need to take cobalt chloride paper and they will be giving, this is white in color actually. So, when we are placing this cobalt chloride paper in cobalt chloride solution and after that if we are drying this, it will become blue in color. Okay. Suppose if I place that by covering with cover slip, slide paper. So, after certain duration of time from the stomata, water is here, a little water is going to be coming out, that is the loss of water by stomata is called transpiration, and here you can observe pink colored patches. Earlier it is blue in color due to the leakage or the release of water from the stomata. The blue color one is going to become pink color. This is the observation what you are going to see during this experiment. The final result is conversion of blue color cobalt chloride paper to the pink color due to the release of water through stomata. That is the inference or result. This is our third experiment. For the preparation and for the slide you are going to get, they have clearly mentioned here, in order to do experiment 3 marks and writing aim, principle, observation and inference 3 marks. Totally 3 plus 3, 6 marks for the third question you are going to score. So, do not write procedure or required things. You need to write only aim, principle, observation, inference or result. Okay. So, now let us concentrate on our last physiology experiments or live experiment, preparation of a paper chromatography in order to find out the pigments present in chloroplast. Okay. Let us do paper chromatography technique. So, this you all know about this is spinach in order to get the chloroplast I got it. Now, by taking this spinach into a mortar and pestle, I am going to add acetone to this, a little bit amount of acetone. You also should do this, and after adding it you need to grind it very gently, then you are going to get a green color solution. Okay. Now, after this, and again I am repeating this, we have to take spinach and add a little amount of acetone in this and grind this in mortar and pestle. Meanwhile, you need to make ready with this chromatography paper. So, now this is a chromatography paper. I made a solvent line and I put one dot over here in order to put this grinded solution here. Now, our ultimate aim is to find the different kinds of pigments present in chloroplast. So, now after this, before you are taking, this is a beaker. In this beaker, I am pouring a buffer solution which is made up of acetone and petroleum ether. Okay. So, in order to transport this pigments present in this solution. Now, I am taking a capillary tube from this whatever the solution we got, from that I am getting by using capillary tube here. A little amount is sufficient. This is acting as standard medium. Now, I am adding at the point of origin. I have added that solution what we have grounded, which is green in color. You can check it out. Now, I am placing this chromatography paper in a mobile medium, a kind of buffer solution. Now, through that, when the buffer is moving along with this, based on their molecular weight and density, the pigments which are present in this solution what we have put also will be moving slowly. After certain duration of time, after the completion of this buffer movement, we need to take this paper out and we need to keep it. Now, you can observe it started moving pigments. A small line you can see moving upside. So, like this, pigments which are based on their molecular weight, they will be moving via buffer solution through this chromatography paper. Now, after the complete movement, we need to take this paper out and we need to keep this for drying. You can see 4 different pigments. At first, this is ascending order, upside is, if you are moving from up to down, the first pigment is carotene, orange color. Next one is yellow color, xanthophyll. Below to that xanthophyll, you can see chlorophyll A, which is bluish green. Then near to the solvent line, you can get chlorophyll B, which is yellowish green. Right now, I am not able to show all those different colors, but once after the movement, if you keep it for drying, definitely you can find 4 different patches. Again, I am saying from the down to up, chlorophyll B, yellowish green, chlorophyll A, bluish green, xanthophyll yellow color, and near to the top position keratin, which is orange color. So with this we have done our all the 3 questions, major questions. First one is family identification, second one is session cutting, and third one is live experiments. So one thing you need to remember, out of these 4 live experiments, they will be giving, if the 20 members are there in examination hall, 5 members will get same kind of live experiments. As a choose basis, they will be giving experiments alternatively. Okay. So what kind of experiment you are getting? Based on that, such kind of aim, principle, procedure is not required. Aim, principle, observation and result you need to write it. Need not to draw any diagram for this session. And most importantly, when you people are doing all these experiments within 1 hour, they will be giving one additional sheet to you, that is for spotters. We have nearly 33 spotters are there, and here each spotter is containing 1 mark. It is giving 1 mark to you. Totally we have 5 spotters. Totally 33 are there in your material manual. So they will be giving D, E, F, G, H. Okay. So they will be placing on certain table and they will be labeled as D, E, F, G, H. So now by observing each spotter what it is, by observation by observing that what you are going to get, how you are going to identify, immediately you need to write. Suppose they have given carrot to you. By observing the color, it is not green in color, and here it is not having any nodes and internodes, so it is not a stem, and it is conical in shape. That is why you can confirm it, it stores the food material, so it is carrot. The first one, only 4 points enough, very easily we made this. So need not to focus on your practical manual, many paragraphs has given. These are 1 mark, carries 1 mark. If you write these 4 points of each spotter based on what you got, it is more than enough. So we have mentioned here for all the spotters according to the TS board manual. So all the spotters, each one 4 points has given. So you need to go through that each spotter, and based on whatever you got in examination, based on whatever you get from that, you need to check and write only 4 points, which gives 1 mark, and mention this immediately. You need to mention about what they have given. So how you identified, for that these points are required. First one, mentioning about spotter and example. Next one, how you found that as so and so. For that these 4 points are very much important. So for all the given spotters, we made it very simplified and we are providing that. Slowly I will move this, you just check it out. So this is the first spotter. Epiphytic vanda. Plant have 2 types of roots, clinging roots which are short, fix the epiphytic plant to branch of tree, and long hanging roots or velamen roots which absorb moisture from the atmosphere, example vanda. Then nodular roots, example arachis. Root system is taproot system, small nodules are found and which are having red color pigment like hemoglobin. Nodules have rhizobium bacteria which fix the atmospheric nitrogen seen in arachis. Brown membranous scale leaves at each node. Stem have apical and axillary buds also with adventitious roots. Then stem tuber, potato, specimen have nodes and internodes, so it is stem. Stem grow under the ground, stores the food material and becomes tuberous. At each node it is containing spots called eye, those are at that eye point axillary buds are developed. Adventitious roots are not seen and on stem like potato. Next one, bulb onion stem is condensed and reduced as disc-like structure. Many fleshy scale leaves are arranged concentrically and adventitious roots developed at the base. Both terminal axillary buds are present. And stem tendrils, for this you can say passiflora as an example, thorns, bougainvillea, offset, pistia, phylloclade, opentia, then phyllodes, acacia melanoxylon, reproductive leaf, bryophyllum, trap leaves, nepenthes, verticil aster inflorescence, leucus, cyathium inflorescence, euphorbia, hypanthodium inflorescence, ficus, berry type of fruit, a fleshy fruit, tomato, hesperidium, it is a kind of fruit, fleshy fruit, citrus, pepo, cucumber, pome, a false fruit, apple, drupe, coconut, nostoc, vegetative filament, spirogyra, vegetative filament, rhizopus, vegetative mycelium, agaricus, a basidiocarp, mercantia, a plant body which is thallus, Funaria, a plant with sporophyte, Pteris, a pteridophytic plant, Selaginella, a pteridophytic plant, Cycas, microsporophyll, Cycas, megasporangium, Pisum, pea plant, Zea mays, corn plant. So, these are the spotters. Out of these many spotters, you are going to get any 5. So, for each spotter, you need to find it, what it is, and for that identification, you need to write 4 points. At last, we have Ricard and Herbarium. Ricard carries, it gives 5 marks to you, and Herbarium for 2 marks. Totally 3 sessions, each 6, 18 marks, and 5 marks per spotters, 18 plus 5, 23, and 7 marks for 5 for Herbarium, and 5 for Ricard and 2 for Herbarium. Totally 30 marks you are going to score in this examination of practical of botany. Okay. These are the diagrams you need to practice. So, these are also you need to practice. It is very mandatory. Everybody without fail you need to draw in your examination paper. It is mandatory. Thank you all. So, hope this video will be very helpful to you. You need not to depend on any of your seniors or teachers. If you go through this video, it is very easy to understand, and you need not to search for any material. All total content regarding botany practical point of view has provided to you. The only thing is you need to prepare for the examination, and here this is very concise and very important points only we have taken. Thank you all.